Welcome everybody to this H3D Foundation webinar. I'm Nicola Elliott-Wong, the program coordinator at the H3D Foundation, and it's my great pleasure to host these sessions. Today we've got Dr. Cloudius Ray Sagandira from CSIR in South Africa, who's going to be speaking about flow chemistry technology. So Dr. Cloudius Ray Sagandira, a principal researcher at the CSIR in South Africa, holds a PhD in chemistry from Nelson Mandela University and is an expert in continuous flow chemistry with over 8 years of experience. Specializing in scalable, sustainable processes for active pharmaceutical ingredients, APIs, he has developed flow processes for more than 15 APIs, forming a cornerstone of API production research in Africa. Through his innovative work, backed by 3 patents and 20 journal publications, Dr. Sagandira is developing modern, sustainable pharmaceutical manufacturing capabilities across Africa, promoting the continent's local API production sector. Dr. Sagandira completed postdoctoral training at Nelson Mandela University, Chimie ParisTech, and DiverChim CDMO in France, and completed an internship at Novartis in Switzerland. Passionate about improving healthcare in low and middle income countries, his leadership and groundbreaking contributions drive sustainable, scalable solutions, significantly enhancing global health accessibility through advanced manufacturing technologies. We're thrilled to have you today, Cloudius. Thank you so much for being here. I'm going to hand straight over to you, and we'll enable the chat function for any comments during the session. And of course, there'll be the Q&A at the end. Thank you so much.
Thank you so much, Nicola, for the kind introduction, and welcome to everyone to this webinar. And I think you have already introduced the topic that I'm going to talk about. It's continuous flow and how we can use it to improve our processes in chemical production as well as API synthesis. So before I get started, I think we just want to talk about this machine on the screen. I think we all know how convenient it is. It's a coffee machine. It's known for its precision, efficiency, and safety. And I would like to pose a question to you before I get started. What if we could run a chemical reaction with the precision of a coffee machine, but more efficient and safer? How would you change the way we make chemicals or medicines? I'll just leave you with that question. So let's get started. I'm going to actually give an introduction, start with the introduction to flow, because of the audience that I have. Probably most of the people are not very familiar with the subject, and I will get into case studies, mainly focusing on the African story. I always like to talk about what we are doing in Africa here. And the biggest question we always get asked is, how can you produce more? Is this technology scalable? So we're just going to talk about scalability of this technology, just a brief introduction to it. Then the last slide will be on how one can set up a flow lab, a cheap flow lab, or a cost-effective flow lab in academic settings. So that will be the last one. And to begin my talk, we need to look at the history of chemistry. And as you might know, over the years, over the decades, there have been so many discoveries in medicine, in different areas of the pharmaceutical value chain. However, the way these medicines are made had never really changed over the decades. And as you can see, it has been anchored on batch manufacturing, which is the use of vessels, flasks, and all that. Nothing changed. Only in the 21st century, flow chemistry technology came onto the platform to try to modernize how chemicals and active pharmaceutical ingredients are made. And why is everyone talking about flow chemistry is the biggest question everyone has. There are social needs. We need to produce chemicals and medicine in a safe way. We need to do it economically. We need to meet the demand that is there. But most importantly, also, we need to take care of our environment. But there's a paradox. We need to produce more, but in a very safe and ecological way. We need to comply also with many regulations and also make sure that we compete as it is not it's not important or it's not wise to change the technology and you can't compete with your competitors. And lastly, we need to produce with less cost and waste. And this is where flow chemistry comes in as a new technology to modernize how chemicals and active pharmaceutical ingredients are made. And the question you might be having is, what is flow chemistry? I always like to start by letting people that flow chemistry is not new chemistry. It's just a new technology to make exactly the same chemistry or to do the same chemistry that you do in batch. So it's referred to many names. Some call it continuous flow chemistry. Some they call it microreactor chemistry. Some they call it just flow chemistry or continuous manufacturing. So it has so many names, but what is it? So it's a way of actually doing chemical reactions in a hollow system or channel or material, typically in microreactors and mesoreactors or tubes, as compared to the batch reactor systems where we use flask that we are all used to. And what is a mesoreactor and what is a microreactor? Microreactor is a reactor with a channel diameter less than 1 millimeter, and mesoreactors mainly used for scale up. They will be having channel diameter of greater than 1 millimeter. And the difference, if you can see here in terms of where you do your chemistry. So in batch, we use a flask at small scale, or at lab scale, a flask like this. At big scale, we use a vessel like this. In flow, we use a chip like this, and these are the channels that I'm talking about. And when we want to scale up, we can use more channels, or we can increase the diameter of the channels. And as you can see here, the difference when you're doing batch, you put your reactants and they tend to react. Then after the reaction, you stop the reaction. But in flow, all the reactants, they are continuously pumped in a temperature controlled or a controlled channel to get the product continuously. It's important to just quickly go through the anatomy of a flow reactor. Since the flow reactor depends on a continuous flow stream, pumps forms the major part of the flow reactor, because the pumps are there to push our reagents through our continuous flow reactor. So we have pumps, and there are so many different types of pumps that can be used. We have syringe pumps at lab scale, and we have HPLC pumps still also at lab scale. We have peristaltic pumps when we want to deal with solids or slurries. At large scale, we have diaphragm pumps that can pump at probably 50 liters per hour or 300 liters per hour when we are scaling up. And we have a mixing zone here. With the mixing zone, we have mixing teas which is important to make sure that reagent A and reagent B mix here before they get into a controlled chamber, which is now the reactor. And with the reactor, we have different types of reactors. We have a column reactor, say we're doing reactions where we have solids. We can pack our palladium on carbon, for example. If you want to do hydrogenation here, then we pump our substrate and hydrogen through this column-packed reactor. We also have tube reactors, as you are seeing here, made of PTFE. Could have been steel, and different materials are used to make these reactors. We also have chip reactors made of different types of materials, from borosilicate to silicon carbide material. And here we can introduce a quench if we need to quench the reaction. If it is a chemical quench, if it is a thermal quench, then what we need to do, when it comes out of the reactor, the reaction stops, because if you are working, say, at 100 degrees, then you put a lower temperature to quench the reaction thermically, then you can quench the reaction that way. And another important part of this system is a pressure regulator. A pressure regulator plays a huge role, because the pressure regulator allows us to pressurize these systems. What does it mean? It means that we are able to do reactions above the boiling point of the solvents that we are using, which is the limitation in most batch setup, because we are limited to the temperature of the boiling point of the solvent, where we have to stick to reflux if we need to heat. So it means if a reaction, you're using a solvent with 50 degrees boiling point, you can easily do the reaction at 150 in continuous flow systems and tend to improve the kinetics. What can you also add on to this? Process analytical tools, which is very important in ensuring that you have quality products and consistent production of material through the continuous flow. So these you can integrate. For example, here I have a benchtop NMR. You can integrate a benchtop NMR, and you can see in real time what you are forming. You can integrate a benchtop IR. You can also, this is another IR also, that you can integrate also into the system to monitor your product in real time. And what are the advantages of flow over batch? There are so many advantages I can talk about, but I'll just try to go through a few of them. I've just highlighted the importance of actually in situ or online monitoring, where you can actually see that your reaction is not doing well in real time and you stop it. You don't need to wait for 3 hours to know that you're not forming what you are supposed to form, or you're forming a lot of byproducts. We talk of safety. It's a huge, huge plus using this technology. So you can do chemistries that are forbidden in batch. The forbidden chemistries can be easily be done in flow, because there's low accumulation of hazardous material that is informed that are formed in situ. In terms of heat and mass transfer, it plays a huge role in making the technology efficient, because there's efficient mixing in these tubes, because they are quite narrow. What you see is there's high surface area to volume ratio that enhances the heat and mass transfer. And as you might know, heat and mass transfer plays a huge role in the kinetics of the reaction. The possibility of pressurizing the system, it means that you can superheat your reactions, resulting in process intensification, drastically reducing the time that you need to do a reaction. In terms of reproducibility, it's one of the best attributes of this technology, because there's precise control of your parameters. Automation, you can integrate, you can easily automate your equipment. At the moment, we There's so much research around self-driving labs where you use artificial intelligence and machine learning to automate and optimize your process through automation. The scale up in flow is much more easier compared to batch, because in batch, when you want to scale up, probably you have seen it, you have done your reaction at say 1 gram scale, you want to go to 100 gram scale. You need to re-optimize your system at every stage of scale up, which means that it's time consuming to actually scale up in batch. However, in flow, it's quite seamless. You don't need to really optimize at every stage of scale up. I'll talk about this point further in the slides to come. Parameter control, I've spoken about it. There's precise parameter control. Miniaturization is a huge benefit of flow. You can easily, that's in very using tiny systems or factories and still get the same amount of productivity. In terms of parameter, there are certain parameters we need to redefine if we're talking of flow chemistry compared to batch. I'll just go through a few that I've selected. Flow rate is a key parameter in flow chemistry, since it affects a lot of things. It affects your productivity, it affects your space-time yield, it also affects your mass and heat transfer, as well as that's a way of also controlling your equivalence or the molarity of the reagents that you are actually putting through. Instead of talking about reaction time as in batch, in flow we talk about residence time, and residence time is the time reagents take to pass through the reactor. So it's dependent on the size of the reactor, which is the size of the total reactor volume, and the speed you are pumping the reagents. And productivity is something that is important in flow, where it is the amount of product that is obtained in an operation time, calculated using Y, which is the mass that you are getting over time. And space-time yield is obtained in an operation within a defined volume of a reactor. What type of chemistry can be done? I think it's a question most people are having. What type of chemistry can you really do? Is it limited? You can do photochemistry in flow. You can do electrochemistry in flow. You can use a microwave heating instead of the traditional way of heating. You can do organometallic chemistry in flow, where you use reactors with a spinning disk or agitation. For example, here is a fluid system where you have spinning disk inside them to provide dynamic mixing. You can do hydrogenation. This is a hydrogenation system where you see this blue thing, it's having water, then hydrogen is formed in situ from electrolysis of water, then you pump it through a reactor, which is pegged with your catalyst to catalyze hydrogenation together with a substrate to perform your reaction. You can also use agitated tube reactors to deal with slurries, because solids are a huge challenge in flow. Nowadays, you can also do continuous flow mechanochemistry, as well as the normal one, which is the solution phase chemistry. So with this technology, you can do virtually all the chemistry that you might want to do, as long as you see the benefit, because there is no need to do a reaction in flow when you can do it in batch, the same conditions and reaction time. There is no need to change the technology. Yes, bringing it home, I think we need to look at how we have been utilizing this technology in our instance as Africa. We have our own challenges as Africa. We bear about 26% of the global disease burden, but we only contribute to 3% of the pharmaceutical production, which means that we heavily rely on imported medicines and active pharmaceutical ingredients. And this usually restricts access to cost-effective medicines and also impairs economical development in the continent. And for sure, there's an obvious need to develop local production capabilities within the continent. However, it doesn't make sense to develop these technologies or to localize production by replicating what is being used in other countries, developed countries, because we become uncompetitive. So the adoption of modern technologies that can enable us to produce APIs at competitive prices with good quality is the way to actually solve our problem. And with me here, a few case studies. And on this slide, I'm not really going to go into the chemistry, but I just want to highlight how powerful the technology is and what it can do. I've worked on a number of drugs that are on these slides, and you can see that, for example, with this in batch, it's a 4-step process, 15 hours long. In continuous flow, we managed to do it in 5.5 minutes, and we telescope the process, which means that from step 1 to step to the last step, we managed to combine all the processes and get 91% overall yield in only 5.5 minutes. If you look at glipizide, 5.5 minutes to do the whole synthesis using continuous flow compared to 18 hours. You look at glycoside, 6 hours long. In continuous flow, we managed to do it in 2.5 minutes. And I can't spend much time on these. If you want to read more about these are the references. We have published this work and also patented it. And what we also did, we worked on these compounds, stuffed in. It was very exciting. I think I have a slide with more detail. 5 steps in continuous flow at lab scale, where we are demonstrating how you can telescope and deal with the different challenges in continuous flow. We reduced the reaction time to 20 minutes over 5 steps compared to the 13 hours and improved the yield. With Tamiflu, this was my PhD work. It was a 12-step process. We did in total. And with this one, for us to actually get to 12 minutes, it was one step that was 8 minutes long, and all the other steps were combined 4 minutes long. So that's how powerful the technology can be. And also we managed to handle AZ chemistry in that system. And if I look at Propofol, we did microwave reaction using Propofol in 22 minutes. Recent work that we did with clonidine, this one is under review. We also reduced the time to 30 minutes, and these are all the references to the work. With this one, I'm not going to actually spend much time on the chemistry. What I just want to show you is how these things can be done, where you're having your reagents here. You determine, you optimize the flow rates, and the flow rates, they control the reaction time, which is the residence time in flow. Then after that, you can telescope if possible. If not, then you have to do them as single steps. So with this one, this is what we did, and I've talked about the results that we got. With this one is the Stafdin. Here we were dealing with handling solids, as well as, for example, here we had a starting material that couldn't dissolve in organic solvents. So what we had to do, we had to immobilize the starting material so that we can be able to do the chemistry. It also meant that after this immobilized packed column, there is less workup that needed to be done, because you tend to get a very clean product. Then you can actually pump it to the next reaction. And this was done from A to Z in a single flow. And we have published this work. With Propofol, we use microwave heating to do this, and I just want to highlight the decarboxylation reaction. It's done at very high temperatures in batch and long residence time and high amount of the base. We managed to do it only in 1.2 minutes. And get 92% isolated yield, and we have also published this work. And probably you might be saying this work resides in the research laboratory here. You know, continuous flow chemistry is actually making a huge impact on the pharmaceutical industry, and with more companies adopting this technology to develop new processes. Obviously, there's that resistance that they can't adopt this technology on the old drugs that they are producing because it's capital intensive. Why destroying something that is working? But with the new drugs, you can see from 2015, this was the first drug that was approved and was made in flow. And you can see that we are having a lot of drugs that are coming in the pipeline at global scale. So this is how important and how this technology is making impact out there. And in terms of scalability, I talked about being seamless. You can, for example, I've just given this one is a conning, these are conning systems. So with the conning systems, you do your reaction at lab scale, where you only have 1 mil reactor at lab scale. When you have figured out your conditions here, done your optimization, and obviously in a 1 mil reactor, you tend to save a lot of reagents and all that. And the optimization is fast. Instead of you spending a month optimizing a process, a month is reduced to a week, for example, because the reaction times are quite small. So from lab scale, for example, from here at lab scale, you can easily go to production scale without re-optimizing anything here. Check the conditions here, you plug them in this reactor, you're already producing tons per year of product. That's how seamless it is. So in terms of scale up, it's very easy, and you tend to get a smaller footprint. The amount of size that you need in flow to produce tons compared to batch, it tends to be reduced. And here is an example of a system where the reactors were 30,000 liters and occupying 650 square meters of space. In flow, it was reduced to 26 liters and only occupying 65 square meters, producing exactly the same production capacity of the drugs. And how can you scale up? I'll just quickly go through it. There are 3 ways that scale up can be done. The first way is where you can put exactly the similar module. You have optimized your reaction in one, for example, in reactor 1, then you just put them in series. Then what it means is you now having the volume of 1 times 3, right? It allows you to pump your reagents at a flow rate that is multiplied by 3. So you tend to increase the productivity. Also here you can do what is called numbering up or parallel reactor modules, where you optimize in two, then you put your reactors in parallel, you pump your materials, then you tend to get your products. And you also can do what is called smart dimensioning. With smart dimensioning, there is a lot of engineering involved to make sure that if you optimize in 3, where you have a channel diameter of 1 millimeter, you do smart dimensioning to 2 millimeter, you need to ensure that you don't lose the mixing efficiency that you were having in 3. So this requires a lot of engineering to make it work. But all in all these systems, they all have their disadvantages. It's not like it's something so simple. For example, with the putting reactors in series, you always have a pressure drop. You realize that if you continue to put your reactors, if you add 10, then the pressure becomes too much, then you can't do that. So there is always a limit. When it comes to numbering up, there is always a limit at how many reactors you can stack up in parallel, because you can't push the reactant evenly in these reactors at a certain height. So you need to be very careful about it. And also with smart dimensioning, I've talked about it. The limitations are that you don't just use a bigger pipe to scale up. It needs a lot of engineering, calculations, and all that to make it work. And how can you actually produce at big scale? You need to combine these methods to actually come up with a process that is very, very good at large scale. And with me, this one is the last part of my talk, where probably some people are very interested to get started on this technology. They don't understand a lot of things, and I'm sure you have seen some equipment there. And as you can imagine, you should know already that those equipments are expensive. But in academic settings, how can you get started without spending a lot of money? So I've just I started by looking. We can start by looking at in what areas can you do or can you use this technology, which is flow chemistry. You can use it in drug discovery, where you do rapid library synthesis, and you also use it in drug discovery, where you can actually quickly synthesize investigational medicinal drugs. In API production, I'm not going to repeat this. I've talked about this throughout my whole presentation. And also you can use also continuous flow in formulation. So apply it across this pharmaceutical value chain to actually improve your process. And you can also include artificial intelligence and machine learning to your systems to actually get better efficiency beyond what is provided by the systems. And in terms of setting up a lab, so I'll just give you a quick, it's not a demo, but a quick way of actually getting started. I'm just calling it a basic startup kit. What do you need? So if you want to quickly start a lab doing flow, what you need to start with are pumps. And the easy option, the cheaper options, is to look for syringe pumps, at least 2, or something that can allow you to pump at least 2 reagents, unless if whatever you are pumping doesn't react when you premix, then it means that you can use 1 pump, you use 1 syringe, you premix your reactants, then you pump them into a flow reactor. Or you can just use a HPLC pump. I know that there are so many HPLC pumps lying around in many people's labs. You can use them to do your reaction. And obviously, you need what we call check valves. Check valves, they make sure that if you are using syringe pumps, you have a flow in one direction, you don't have backflow, which is important in terms of the system being consistent. So these are the types of check valves that you can buy. They're not expensive. And what else do you need? Obviously, you need to join reactant A, reactant B. You need mixing T's. It's just a T where you just join your reactant streams. You need to buy these. They're also cheap. Not that cheap, but they're cheap. And in terms of the reactor, I always encourage people who are starting, especially in academic setting, to go for PTFE tubing, because in flow, when you are learning, you tend to block reactors. And if you are to buy one glass chip, which is having probably 1.5 mil, can cost you 1,500 US, a glass chip. And you have these new students starting on a project and makes some mistakes and just drops it, and that's the end. So with this tubing, you can get it, I think, a row of 30 meters. Probably you get it at 1,000. You find it at Sigma, 1,000 or 2,000 rands. Then you can use it to make the size of the reactor that you want, depending on what you want. And you need to know the diameter that you want. If you're doing chemistry needing solids, you need this type of a reactor. You can buy any cartridge that you can see and pack your stuff here. In terms of temperature, we go for the cheap stuff. We all have these hot plates and the oil baths. Then you can dip our things in the oil bath, then we control temperature that way. Back pressure regulators, very important if you want to go beyond the reaction, the temperature of the solvent. And there you are, you can now collect your products. And I think you can see that it's easy to easily set up this reaction. And the message I can just leave you with is let's go with the flow and try to explore how it can also reduce the burden that we have and reduce the pressure. I know some people, they come from medchem background. Imagine if you can easily synthesize a library in a day, 1,000 compounds, 100 compounds using this technology. It will be massive. And here is just a link where you can actually buy all these small things I have just mentioned. And to complete my talk, I just wanted to introduce our facility at CSIR, which we call the Future Pharma Facility, where we are developing technologies for local production of APIs. We do a lot of collaboration with industry, local, and also international. We are mandated by the government to do this work and also support the industry in actually finding ways of localizing continuous flow manufacturing. So here we have our new batch lab, so many equipment, and we have lab scale flow chem lab. I think we have a lot of equipment, different chemistry that you can do, from Verbotech, your Unixes, your SGDs, and your benchtop NMRs, your IRs, and at pilot scale. We have recently installed these pilot scale equipment that can allow us to produce up to 400 kilograms of product per hour using only a 2.5 liter reactor and pilot scale. That's how powerful the technology is. And we also have a smaller one here where you can actually produce up to 40 kilograms of product in a 20 liter, in a 200 mil reactor volume, actually, not And this is a industrial rotor vapor there that we are currently using to push our work in the pilot plant. And if ever you are willing to collaborate, we also do support student projects a lot. And we also have a lot of students that get involved in our projects and get to actually work in our laboratories at CSIR, where we collaborate with on various projects, and we try to teach students and also the industry on how to do the chemistry in flow from lab scale up to pilot scale. So if ever you thinking of venturing into this, feel free to get hold of us and we can discuss how we can collaborate on different projects or even projects beyond API production, any chemistry that you want to do and you think that flow chemistry can make it easier, then we can talk. All right, guys, thank you so much for the for listening to my presentation, and I would really like to thank Nicola and the H3D Foundation for inviting me to share this work that I'm really passionate about, and I really hope a lot of people will be interested in also trying this technology in different areas of their research and try to improve their processes and lessen the work they do. Thank you so much. Thank you, Nicola. Over to you.
Thank you, Claudius. What a fascinating presentation. And I'm sure we all get excited by the concept of optimizing workflows and having less time spent in, the process of it and actually getting through. So that slide, particularly on that, when it showed how much you reduced the time spent on certain processes, was extremely satisfying. And there's already something coming through on the chat. And everyone, if you want to ask a question, the mics have been enabled, so you're welcome to just raise a hand and then unmute yourself when it's your turn. So this is a question from Buyasane. Good day. I'm a PhD student at Rhodes in the biochemistry department doing research on drug discovery and cancer. I've got two important questions. Number one, what attracted you to this field? Number two, looking to the future, what are the key challenges facing flow and scale-up chemistry with respect to the pharmaceutical industries? What fantastic questions. Claudius, are you up to answering those?
All right. In terms of the first one, thank you so much for that. For those two questions, very good questions. For the first one, in terms of attraction, I'm really passionate about improving health access in developing countries, and growing up, we see the challenges that we face. We lose people just because we don't have access to the medicines. We lose people because the medicines are too expensive, right? So how can we solve the challenges that we are facing as Africans? I think we have the solutions and we have the skills. And that need of actually actually making sure that we improve access, that's the thing that really attracted me to continuous flow technology. And I was very fortunate to actually be trained by one of the leading researchers in the field. And I always mention it because, he taught me a lot of things. Professor Watts down in Nelson Mandela. We did a lot of work together and he taught me a lot. And that cemented my passion, the technology. And I realized how powerful the technology is. And for the second question, it's very key. And I've been trying to push this work and for the past, I think, 8 years. Also here in Africa, I've done a lot of work in Africa, developed few, if not probably the most targets that have been developed in flow. I can say, I think most of them I was involved as the lead. And I had also an opportunity to go to France, where I was opening a flow chem lab, where in 1 year we developed 3 processes for 3A APIs. You always see the challenge with this technology is that funding is one of the challenges, and also the skills. We are trying to develop a whole industry here, but do we have enough people who are going to drive the industry? If we don't have enough people who are going to drive the industry forward, it's not going to be really easy. And yeah, we appreciate that we have government that is really supporting, because the government is really putting a lot of weight on this. But obviously, they would really love to see some products, for example, at pilot scale formed. But you running a pilot scale production of 400 kilograms of product, right, to just demonstrate, it's very costly labor-wise and also in terms of chemicals. And after that, what you do, you flush it down the drain. You have to ask for someone to come and dispose that material, which will be a waste. So it's important to actually have industry collaboration that will actually ensure that they will take up these technologies that we are developing, which is a huge challenge for us here in Africa, because we are trying to establish an industry here, trying to support an industry that is not well established. It's unlike if you're in Europe, you can get these projects that are industry specific. A client comes to you and say, can you produce this for me? That one is a different discussion altogether. But at the moment, this is the challenge that we are facing. We are very positive, and we are very positive on what the technology can help us achieve and improve the access to medicine. Thanks.
Thank you so much. Are there any other questions? Tembela, off you go. Go for it.
Thanks, Doc, for the insightful talk. I wanted to ask about your stavudine synthesis, because I saw there that you have your peg bed reactor.
Yeah.
I wanted to know, is it practical if you want to take it to pilot scale or production? Thank you.
Okay. So with peg bed reactors, right, those are not my favorite. They're very difficult. You can deal with them because you can do what we call, you have about, say, 5 cartridges pegged with the reactant. Then obviously you have to use inline analysis integrated in the system. Then you have to use a reagent switch. Then when this first, say the first reactor that is pegged is used up, the system tells the reactor, or the system notifies the reactor, and you switch to the second one automatically. So you can do it, and you can also regenerate them also that way. But it's very time consuming, but it's doable. What I would prefer myself is trying, in that case, now that I have a different equipment that can help, that can handle slurries, which are solids, what I would do is I would rather do them as slurries, and it's very easy to scale up that way. So if you are having a spinning disc type of a reactor, if you are having an agitated reactor, but what usually happens, the flow labs, you can't have everything that you need. So you need to be very creative to be able to actually do the work that you need to do at that point in time. But if I'm given another opportunity now, I'll do it way better than that, and even at a better scale. Thanks.
Thanks.
Okay. Thank you. I've got a question on the chat from a former colleague of yours from NMU, Francis Mate. My question is upscaling from lab to pilot. You mentioned that we might need some engineering assistance in sizing up. Since the majority in this field are chemists, are there perhaps user-friendly tools that can be used to simulate the process?
Indeed, it's quite unfortunate that actually this technology is actually mainly driven by chemists. But one thing I saw in Europe, you go in the USA, they're actually engineers that actually do continuous flow technology. So they are well equipped to actually do this type of work. And in terms of sizing up, what people usually do, they usually go for commercial equipment. So if you buy a lab scale equipment from Chemtrix where you're doing it microliter, then when you need to size up to production scale, you need to buy an Chemtrix, for example, at that scale. Then you don't need a lot of engineering anyway. But if you are building your own, I know Francis quite well because he's on the side of actually doing some engineering stuff and trying to build his own systems. So that that requires a lot of effort and knowledge around engineering principles and all that. And probably at this point in time, you have to try to use open source technologies out there to try to find help on how to do the scale up in a manageable way. Otherwise, it's very difficult because these commercial companies, they don't show you how it's done and how they can size up in a smart way. Thanks.
Thank you. Before going to our final question, I just want to tell everyone there's some links in the chat, including a link to the feedback form for the webinar series. So feel free to go and drop your comments in there. Kanyisile, over to you for our final question.
Hi, Dr. Saga. Thank you so much for the great presentation. My question is it kind of comes across like an economy type of question. So you mentioned it a little bit, but I wanted to know that, can you modify simple reactors to do reactions for photochemistry, electrochemistry, or do you have to buy specific reactors for that specific type of reaction? Thank you.
The simple answer is, if you want to do it quickly, you have to buy the reactors. You have to buy the reactors from the suppliers. But also, there is an opportunity that you develop yours because the principles are just straightforward. You develop yours like the one I've just showed them. You develop yours where you are doing a homemade system. And this is how these commercial reactors started. They started in the labs and they tested them and do a lot of things. So you can develop yours. You can use 3D printing. We also have those capabilities where you do 3D printing of your reactors. Then you can actually develop your own reactors. But obviously, that will also require a lot of engineering behind it and a lot of understanding of what you're trying to do. So a mere chemist, organic chemist, might really struggle to get it going, but if you're smart enough, you will get it to the end. Thanks.
Right, I think that brings us to the end of our session. Cloudius, thank you so much for your time and energy and expertise and passion. It was a fantastic presentation and some really great Q&A responses. Thank you. And thank you to everyone who's joined us today. I think we've got one more coming up in our webinar series for this quarter. And I look forward to seeing you there. And until then, I wish you a wonderful week further. Thanks so much, everyone. Bye.
Thank you.
Thank you.
Bye.
Bye.